Okay.
Okay, we're live here in Eugene, Oregon with the Cascade Mycological Society's invited speaker for December 2022, Dr. Peter Kennedy. Um, Dr. Kennedy is a professor at the University of Minnesota, and he's here to discuss his lab's recent explorations of the specificity of associations between trees and their ectomycorrhizal fungi. The work is focused primarily on two related fungal genera that are abundant in our region in the Pacific Northwest, Suillus and Rhizopogon. Much of Dr. Kennedy's research combines pairing field surveys of tree fungus pairings with experimental bioassays, testing patterns of specificity in different host environments. And understanding these patterns of host specificity is not only helpful in mushroom foraging and identification, but may also be increasingly important for understanding how forest diversity will respond to changing climate conditions. Dr. Peter Kennedy is a professor at the University of Minnesota. He did his undergraduate at Evergreen State College in Washington State and his PhD at the University of California, Berkeley. Please give a warm welcome in your own way to Dr. Peter Kennedy. Dr. Kennedy?
All right. Well, thank you so much, Chris, and thank you to the Mycological Society for this invitation. I am from the West Coast originally, and so I've spent a lot of time looking for fungi in your neck of the woods, and it's really something that got started there, even though I'm now here in Minnesota. And so I'm glad to be able to tell you a little bit about the research that we've been doing in my lab. And so I'm going to go ahead and jump kind of right in, if that's okay. So I think, this picture kind of encapsulates to me what a lot of folks think of when they think about fungi, and maybe why some of you have joined the Cascade Mycological Society. So, many different types of mushrooms in forests. They are absolutely beautiful, each in their own way. And, trying to understand when I started thinking about fungi and learning about fungi, where they fruit and why they fruit and sort of what time of year they fruit. These were all questions that were sort of on my mind. And one of the big things that stands out, which I'm sorry, I don't know kind of at what level you have background. And so I'm going to start assuming that some of you are relatively new to thinking about fungi. But for some of you who don't know, there's actually a lot of the fungi that show up in forests that have this special connection to, in this case, just going to see if I can make the slides cooperate. Why is that not? Oh, interesting. Let me try one quick thing where maybe if I go into this mode. Sorry about that. So hopefully you can still see everything. You can see that there is this close association between the mushrooms that we like to find and collect in the forest. And actually, if you were able to dig down in the soil, you'd look at their mycelium. And if you followed that mycelium, it would all the way take itself back to a tree root in many cases. And it's this interaction between fungi and trees that really has been the focus of a lot of the research in my lab and what I'll do some talking about today. And so thinking about that, this is another picture of what this relationship looks like. So if you were walking through the forest and you saw one little teeny pine seedling sticking its head up above the soil, if you were to look down into the soil, you might actually see something that looks a lot like this. And so you can see that there is a whole bunch of white mycelium. So that's all the fungus that's growing out into the soil. And then there's the seedling, and there's these little white dots on those roots. And what's happening is there's this interaction between the trees, which are making sugars from photosynthesis, and so they're passing that below ground onto these fungi, and in return the fungi are out into the soils. Can you see, Chris?
We're not seeing a picture. We're just seeing the sketch with a blank.
Sorry, let me see if I can.
Do you need to be in slideshow mode? And you might have to reshare to do that.
Yeah, let me try and reshare real quick. Sorry, let me try one more time here. I think this is gonna work. How about that?
There.
Is that? Can you see the slide?
Yes, we can see a slide now.
Excellent. Okay, so sorry about that. So this would be a seedling that you can see above ground, and then below ground you can see all this mycelium of these fungi. that are hooked up with these tree roots. And what's happening here is you've got this interaction between plants that are able to make sugars from photosynthesis, and they are passing that below ground. And in return, the fungi are helping these plants get access to lots of different things, but particularly nutrients, things like nitrogen, phosphorus, that are hard for the plants to get access to on their own. And so the name for this symbiosis, and what we study a lot in my lab, is these mycorrhizal associations. And so myco is fungus and rhiza is root. And so it's actually a physical union of the plant and the fungus that defines this symbiosis. So in terms of thinking about these mycorrhizas and plant growth, this is a picture actually that was an experiment done in Oregon back in the 1980s. And it's sort of one of these classic pictures where you have plants that have access to these mycorrhizal fungi on the roots, and then you can basically grow the same plants where they don't have access to those same fungi. And what you can see is some plants that are much bigger on the left side there. So they have those, what we refer to as ectomycorrhizal fungi, versus the plants on the right don't have those fungi. It's actually a little more complicated than that. So when you can take the same fungus and grow it on lots of different plant species, you can see actually there's a real range of responses in terms of the amount and size that the plant can grow to with and without that fungus there. But there's been some studies that have kind of synthesized this broader literature. And what they've been able to show pretty convincingly, to me anyways, is that you can see across all these different groups of plants. So what you're seeing here from left to right on this x-axis is the different types of plants. And then what your y-axis is 0 means it would be the same response if the fungus was with the plant, and if it was below, then the plant would actually be growing less, and if it was above, it would be growing more. And in almost every case, you can see that there is sort of that solid, horizontal line there suggests, there's about a 60% increase for these grasses when they have these mycorrhizal associations. And this is true of lots of different types of plants. There's some context to the environment and those conditions that matter a little bit. So if you have a lot of nitrogen that's been added into the ecosystem that you're looking at, the benefit of having these fungi might be a little bit lower than it is in areas where there's less added nitrogen. But overall, it really looks like there is a positive benefit to having these mycorrhizal fungi helping plants grow. So exactly how they do it is sort of interesting. And so there's some background here thinking about what these fungi do. So the fungi can basically grow out into the soil and really extend the root system. And they can do it in a way where they're able to cover more of the root system and grow into, sort of further parts away from the roots and access more soil. And then because they're very small, they can actually get into soil pores that themselves are, in this case, not accessible to the roots as well. So both of those can be ways that the plants can benefit from access to greater resources. It's like I just said, they can get into those sort of smaller pores, and they can get access to those last bits of water that are there in, say, dry soils in particular. And then they have some enzymes that plants don't have that allow them to go out into soil and basically break down organic matter into simpler forms that then can be taken up and traded with the plants for the sugars that the plants are making from photosynthesis. So this is a really important symbiosis. There's actually a lot of interesting ecology behind it. And so we know, for example, that a lot of what's happening above ground in terms of the plant community, how diverse that community is, how it goes through different stages of, say, succession from a major disturbance that sort of wipes out, say, your forest to one that really becomes an old growth or a climax forest. All of this can be strongly influenced by the presence of mycorrhizal fungi. And so that's been sort of a fun way and sort of a way to re-envision thinking about forests and how important things are happening below ground for what things look like above ground. These play a really important role in the movement of nutrients through these ecosystems. So whether it's carbon and moving through the ecosystem, and of course, this is something we care a lot about in the context of the current global change that we're experiencing now, particularly with climatic change, but also major nutrients that are a key part of the life of all ecosystems, things like nitrogen, phosphorus. All of these are really mediated in major ways by these mycorrhizal symbioses. Of course, another big thing is they are a big part of food webs, and that's both the mushrooms that we like to eat, of course, but also the mycelium is a huge important source of, in this case, food for lots of what we call the brown food web, especially in soil is what I mean there. So they kind of make up the basis for lots of the food web, and that can actually then trickle up into some fascinating stories of some of the fungi that are involved as particularly truffles and feeding the diets of lots of different animals in old growth forests in the Northwest as well. And then the last thing that's, I think, interesting that, again, maybe some of you do and some of you don't know about this, but it seems like the symbiosis or the interaction between fungi and plants, this mycorrhizal symbiosis, was really kind of a key step in allowing plants to colonize lots of land. So plants, of course, started as algae out in the oceans, and then as they moved into terrestrial environments, these symbioses with fungi really seemed to be a key factor that facilitated their success in terms of getting access to nutrients in sort of early land conditions. So lots of, for me, interesting reasons to think about these mycorrhizal symbioses. But today what I want to do is I really want to focus on one particular genus. I realized when I was putting the talk together that I've got plenty in here just talking about Suillus. I'm happy to answer and talk to you about the work that we've done on Rhizopogon as well. But today I think I'll really focus mostly on the genus Suillus. And so Suillus are a group of, in this case, forest-associated mushrooms. The common name for them is the slippery jacks. So you find these in conifer forests. They're cosmopolitanly distributed, meaning they're basically in all parts naturally of the Northern Hemisphere. So we have them in the Americas, they're present in Europe, they're present in Asia. They've been introduced into parts of South America as well, but really they're mainly sort of a temperate and boreal associated fungal group. So they don't really exist in the tropics per se. They're relatively easy to identify. And what I mean by that is, in terms of recognition, I think a lot of folks who are just learning mushrooms, kind of learning what is a slippery jack, is one of the first ones that I think can be pretty differentiable. And I'll talk about that relative to some other boletes or pored fungi as well, but they're common. So you see them a lot, and that's true certainly in the Pacific Northwest as it is here in Minnesota. They're important ecologically, so they play a pretty key role in this case, various aspects of carbon and nitrogen cycling, response to disturbance. Economically, they're a key part of actually helping with forest establishment, and they are actually harvested and eaten, although they can't be grown in cultivation, so they're not really harvested in a commercial way, in a major way. The thing that's exciting for me as someone who studies mycology but really likes to do experiments to help us better understand the kind of ecological processes that we're interested in is that we can take these mushrooms and we can get access to their spores, and we can use those spores to do experiments with seedlings that we refer to as bioassays. And so that's what, when Chris mentioned that in the introduction, I'm going to talk to you about some of these experimental plantings that we do to help us better understand how these fungi interact with different types of trees. So I'm going to start off with what is a Suillus. And so what is a Suillus? Well, it is, I think these are some kind of large and defining features. So it's morphologically, it's a bolete. So that means that it has pores on its undersurface rather than things like gills or teeth. And it has pores that have an irregular shape to them. So that's one thing that helps differentiate it from some boletes and others that look like it in this group. And the big thing, and the reason why I think it gets the name slippery jacks, is because it often has a quite slimy or viscid cap. So this is a close-up of some of these pores. And so if you look closely at the pore surface, what you see is they really have sort of an irregular shape. So they're not tubes that are perfectly circular, which is much more common on the boletes. And in some Suillus, they're actually pretty big size pores, but in others, they can be relatively tight. And a lot of that depends on the age of the mushroom. But that's one feature that can help you when you're looking at, say, a slippery jack versus a bolete. So many irregular pores. And then this slime layer, Suillus viscidus is the species here, is really another distinguishing feature. And so they often have slime on their cap. and it can be impressively slimy. So if you've ever been out in the woods, and certainly, there are plenty of these mushrooms that exist. Suillus pungens is a West Coast species that you might see when you're out towards the Oregon coast, but they can be really, really thick and covered in this clear, almost completely tacky liquid-like structure. But they can also be quite dry. So depending on the habitat that you're in, Suillus lakei, that's another Doug fir associated Suillus species that you find, I think, a fair amount in your neck of the woods, hopefully. And it has a dry surface, but it has fibers there on the cap. And this is a mushroom that you don't have in Western North America, although there's some relatives that are present in some of the high elevation locations. But this is Suillus americanus. It's called the chicken fat mushroom. It is one that's eaten with some frequency here in the Midwest and in the East, although there's a few other Suillus that are more preferable. But these ones also have a relatively dry cap surface. So that's a little bit of background of what they look like. In terms of how we know them scientifically, a couple of folks who did a lot of work on this. So Alexander Smith, who is a famous mycologist from the Midwest, but spent a lot of time in Western North America. He's a professor at the University of Michigan. And Harry Tiers, who's a longtime professor down in the Bay Area. They worked on defining the American species of Suillus. And so we think we probably have something like 130 to 40 species of Suillus that are present globally. We have a lot of representation of Suillus, particularly in Western North America. But I was fortunate to work with a fantastic postdoc who's now a professor at the University of Hawaii. His name's Nu Nguyen. And Nu and I and Elsa and Tom published a paper where we went back and we used some molecular analysis to kind of revisit the species concepts that Smith and Tiers had put together in their major monograph. And we did find that the general patterns that Smith and Tiers put together made sense, and a lot of it has to do with which types of trees they associate with. But we also found that a lot of the names needed to change. We took a lot of European names of, in this case, slippery jack mushrooms in Europe. And then when we sequenced them, we found out that they had actually some very different, in this case, molecular identity than the ones that were present here in North America. And so we had to update and change some of the names. So not going to dig sort of in super deep detail into the taxonomy, but I think if you're interested in this group of mushrooms, these are a couple of good resources to start with. In terms of the species, though, it does get tough. They can be hard to identify. And so one of the big things, of course, is which host they're associated with, and I'll talk about that more there. But of the macroscopic characters, there's actually a lot of variation. And so you can get ones that are side by side. Sometimes they have that viscid cap, sometimes they don't. You can see they change a lot in terms of color as they age. So sometimes they're, present and they can look completely pale on top, and then you come back and they can be completely orange, or many other colors are speckled as well. They also can get pretty slimy pretty quickly. They seem to be a favorite habitat of lots of insects. And so if you've collected Suillus and you've forgotten about collecting Suillus, it's very likely that you will end up with a pile of mush, unfortunately, that can be hard to identify when it looks like this. They do have a few features. So you have a species that is called, it's associated with large trees in the Cascades. Suillus grevillei is the name you'd find it in mushroom guides, but it actually has a new name. The correct name for the North American version of this is Suillus clintoniensis. And Suillus clintoniensis has this cool chemical reaction. So if you take potassium hydroxide, a common identifier compound that you could put onto mushrooms to see if their color changes, and you cut one of these open, I'm going to run the slide here. Hopefully you can see it. You can see that there's this pink flash that happens. So it happens pretty quickly, but hopefully you saw that there, that the KOH reacts very quickly and it sort of turns green. So that's one of the characters, but there's not a whole lot of these. So it can be somewhat tricky to identify Suillus down to the species level. But in my opinion, it's definitely a fun effort. And I think that the diversity, particularly on the West Coast, is pretty manageable. And so hopefully you guys are excited by this talk to go out and take a look at some slippery jacks and see if you can put some names on them. Some other cool characters about Suillus, sometimes they have veils, and that can be really important, whether they have a veil or whether they don't. So that's that ring that is left on the oh goodness, on the stipe, but the veil sometimes will cover the pore surface. Whether they have these glandular dots on the stipe can be important. Whether they have fibers on the cap is another factor that matters. Again, color changes, although color changes really, that one sometimes works. There's a real common Suillus serrilescens, the one you have out on the West Coast with Doug Fir, that has a bluing to it. And then sometimes they have these interesting sort of glandular kind of droplets that can show up. In terms of, you know, these are relatively minor features. Is these milk droplets, they're hard to see. It really depends on humidity conditions. The sour taste, again, something that's not consistent, and the bluing really is, again, not a particularly helpful character in the same way that it is with, say, the genus Boletus, where really some of the staining reactions can be quite important. So that's kind of a broad background, but I wanted to introduce you to the group of mushrooms that I'm going to spend the rest of the talk focusing on. So this is a phylogenetic tree. And so for those of you who are familiar with these kinds of analyses, basically what you do is you grind up samples of each of your mushrooms, and then you sequence a fragment of their DNA, and you take that fragment and you line it up across all the different mushrooms that you're looking at, and then you reconstruct how they're related to each other. And so in this case, these mushrooms, or these samples that are closer together in these trees, have, in this case, a closer evolutionary history than those that are further apart. And so Annette Kretzer published this paper back in the mid-1990s. And the key thing that I'm trying to show you here is that for the genus Suillus, a lot of what we know about it has to do with these host associations. So a lot of these mushrooms, in fact, are well known to associate with just specific groups of, in this case, conifers. And so we've got pines, and we've got two versions of pines. We've got hard and soft pines, or the 2 and 3 needle versus the 5 needle pines. We've got Douglas fir, which of course is a big tree in your neck of the woods. So Suillus serrilescens, Suillus lakea, as I mentioned before. And then if you go up into higher elevations, you'll see larch in the forests in the Cascades. And we have a lot of larch, one species, but a lot of larch forests here in Minnesota. And they have a whole set of Suillus species as well. But I remember looking at this tree early on and thinking, there's this one kind of weird and interesting exception, and it's this Suillus savarius, and it's got a hardwood association. So Suillus is notoriously known with the pine trees, but there's this one species that looks like it hangs out with hardwoods. And this is a picture of what it looks like. It's got this beautiful kind of apricot color to it. And so when I first moved out to Minnesota, I knew Suillus savarius was here. It wasn't on the West Coast where I was before I started working in Minnesota. And so I really wanted to learn more about whether this was this right, or was this, something that maybe in mixed forest it was actually hiding out with some conifer trees that we just weren't paying attention to. So luckily, my first PhD student, a really fantastic scientist named Lotus Lofgren, did a bunch of work on this for her PhD thesis, and Nguyen was actually a postdoc in the lab at the time. So Lotus and Nhu took this on, and it was one of the, again, first projects that we really jumped in on. So, I think for mycologists, if you can be studying one of the 100 cool mushrooms, I think that's a good sign that you're on track. And so I actually found this book when I got here to the Midwest, and Suillus savarius was in the book. And so it says here, one of the defining features of the genus Suillus is a mycorrhizal association with conifers. Here's one that does not always associate with conifers, often found under quaking or big-tooth aspen without conifers in the vicinity. So this one really seemed like a kind of fun puzzle or anomaly to understand what was going on with these different types of host association. So I'm going to walk you through this study. So basically, we wanted to ask ourselves, are we seeing Suillus savarius associated with the roots, right? So that's where that symbiosis is actually happening on hardwood hosts in the field. And then we're going to kind of take this and do some experiments to pull this apart. And so which host does Suillus savarius associate with in these seedling, in these experimental plantings that we can do back in growth chambers or in greenhouses in the laboratory? And then how can we maybe link this into the ecology of those forests? And so I'm going to circle back and try and help make sense of what we think is going on with this pattern. So this is the state of Minnesota. I am located currently right down here in this part of the state. And one of the nice things about the state, in addition to the many lakes, is that they have a number of scientific and natural areas. There's actually about 170 of these where they preserved interesting landscapes or unique geologic features in Minnesota. And so when I first got here, one of the mycologists said, well, I know of a spot where Suillus sabarius is. It's at Lake Alexander, SNA, so you should go out there. So my postdoc and I drove out there in 2014 during the first mushroom season, and we were able to find a collection of Suillus sabarius, and it was growing in an area. So this is actually the site where the road is. So we hiked in and we found it, and there was only hardwood trees, and particularly there was some red oak trees, Quercus rubra, that were very nearby. In this whole area, there was one kind of medium-sized Pinus strobus tree, and we were finding Suillus americanus under that Pinus strobus tree. We knew that was a pine associate. And then we went back in 2015 and we scoured the whole forest again, and we found one other collection of Suillus sabarius, and it was associated with, again, most closely to Quercus rubra, but a long ways away from where this pine was, probably well outside the range of where the roots of this pine tree was. So that seemed like, interesting. We should try to look harder, and we should try to figure it out. So we knew it was that mushroom, but one of the things that we decided to do right away was take soil cores. And so directly over where we harvested the mushroom, we took a soil core. We took 6 of these in this forest at Lake Alexander, and we used these DNA techniques to identify both the plant and the fungus. So we pulled out all the root tips that were ectomycorrhizal and had this kind of Suillus morphology, so nice kind of white and fluffy. We sequenced them, and what we saw was that we could get Suilloid tips, again, ones that looked like Suillus, in 4 of the 6 cores directly under where we were finding Suillus sabarius. And we were able to get, in this case, confirmation that those root tips were colonized by Suillus sabarius using our fungal DNA barcodes. And then we don't have the greatest evidence here. We were actually able to work at another site in Indiana to kind of nail this down a bit further. But we did find that when we sequenced the roots, and at least one of those cores, and it was a Suillus sabarius colonized root tip, it belonged to Quercus rubra. So that was a big deal, because that for us really established that it was actually Suillus sabarius was associating with hardwood hosts in a field setting. So that was an exciting first step. And so we thought, okay, we've got the mushrooms, we can get their spores. Let's go into the lab and see if we can put those spores onto the roots of different tree types and really nail this down. So this was some work that Lotus Lofgren did. So we did a bioassay. So we collected all those spores and we added them into 3 different host settings. So we had 2 different types of oak trees. We had red oaks and white oaks, and then we had some aspens. And we waited a long time, and we harvested actually at 2 different times, after 3 months and after 6 months, and we got 0 colonization. So there was no mycorrhizal colonization of Suillus sabarius on any of the trees that we were looking at. So this left us kind of scratching our heads. So we find it in the field on oak trees. There's reports of it on aspen. We didn't see it here. But how is it there if when we take the spores, we can't get it to germinate any of those oak trees in otherwise ideal growing conditions back in the lab? So we decided to look back at where, that should say Suillus sabarius, my apologies, but where Suillus sabarius sat within the phylogeny of all of the slippery jay. And what we saw was that it actually, Suillus sabarius, is closely related to pine-associated mushrooms, particularly 5-needle pine mushrooms, which in this case, Suillus and 5-needle pines, the common host that we have in Minnesota is white pine, eastern white pine. You have western white pine up in the mountains. You also have, what is the one with the very big cones? Pinus lambertiana. I'm blanking on the common name for it. Sugar pine, is that what it is? Anyways, there are some 5-needle pines in Oregon, but they're not so common as they are.
Ponderosa pine?
Ponderosa pine is a 2-needle. The length of the cone is massive. It's Pinus lambertiana, and I'm just sorry, not coming to me, but
Sugar
it will.
pine.
Sugar pine, thank you. Could be. And So yeah, anyways, so we thought, okay, well, maybe that's something interesting. And so we kind of speculated, and our speculation was, could Suillus sabarius spores, maybe what they do is they germinate if pines are present. Pine roots maybe have some germination cue, and then they can grow out from those pine roots and they can colonize hardwood hosts that are growing in the immediate vicinity, but they do it from mycelium, not from spore germination. So Lotus worked really hard and set up this cool experiment where basically what we did was we added some different species of Suillus. And so we had that Suillus americanus, which we knew is associated with, again, our eastern white pine, which is Pinus strobus. And we had Suillus sabarius, which again, we'd found on Quercus rubra. And so we planted these combinations of these little seedlings in these pots where we grew with just the oaks, with just the pines, or a mix of the oak and the pines. And we did that with Suillus sabarius or Suillus americanus. And then we actually waited a while. And in the meantime, what we decided to do was we decided to cut off what we thought maybe was the original host and see if that made any difference. And we only did this with Suillus sabarius. We didn't end up doing this with Suillus americanus, but this was the design of that experiment. So these are the results that we have here. So we're focusing just on the left graph here on the pine trees. And so what we're focusing on is how much of the pine root system is colonized. And so this is, half the root system is colonized down to no colonization. And we do this in a way where, again, we don't usually expect to get 100% colonization. And so this is kind of what normal levels look like for us over here. But we see that in this case, the Suillus americanus and Suillus sabarius both colonize the pine pretty well. There's lots and lots of mycorrhizal roots of both of those two species in the pine-only pots. But neither one of these two species colonizes, in this case, any of the roots of the oaks when we grow two oaks in the pot. So this really suggests that when the hosts are alone, we only see germination of the spores of Suillus sabarius in the presence of pine. And I guess we could say the same thing for Suillus americanus, but of course sabarius is the species we're most interested in. So now let's look at the mixed pots, right? So we've got both an oak and a pine present, and we're going to focus on the two different hosts, okay? So if we have the pine present and the oak present, but we look at just the pine roots, we see again there's reasonable colonization of the two different Suillus species. Suillus americanus a little higher than Suillus sabarius, but there's definitely colonization of Suillus sabarius. Yet in the exact same pot right next door, after 5 months, we don't see any colonization at all on the oak roots. So it really looks like this Suillus species is very specific to just the pines. And so again, this kind of had us really scratching our heads. Well, we would think maybe it would have jumped over onto the oaks. And that's why we can maybe see it potentially in other settings, but we aren't seeing that. So we waited around for 7 months. We gave it 2 more months, and we did that little experiment where we left some of the pines alive, and then in others we got rid of the pines. Now the colonization is a lot lower in terms of the overall absolute amount. So we're looking at 4% here, but again, it has to do with part of the ways that we quantify these mycorrhizal roots. We don't count each individual tip, we do it on a weight basis. And if you look at a picture of this, there's actually lots of good colonization. So these white blotches are covered in lots of little root tips, but oak roots are kind of teeny and don't weigh much. And there's a whole lot of oak roots in these pots. If you grow a seedling for 7 months in a pot, they really do produce a lot of roots. But as you can see, there's good colonization here. And we actually see that, yes, Suillus sabarius has colonized these oaks in both of these two settings. So this is interesting. This suggests that Suillus sabarius does colonize Quercus, but only after a long time, 7 months in this case, and with pines originally present. That seems to be a key requirement. So for this first study, the main conclusions that you can take home are that unlike other slippery jacks, it really does look like Suillus can associate with multiple hosts. In this case, both the pines that we saw it associate with Suillus sabarius and with, in this case, the oaks. What we think, though, is that this is an interesting quirk of biology. And so we think the current association with hardwood hosts may reflect what we refer to as a ghost of mycorrhizal past. I didn't mean to put this in the holiday timing of this talk, but maybe it fits well with with the timing. And what we mean by that is that we think that this species can only start on pine trees, meaning that those pines have special chemicals in their roots that allow the spores to germinate. But eventually they all got cut out of Minnesota. Turns out, if you didn't know, Weyerhaeuser, the company that's famous for owning lots of land and timber in the western United States, started in Minnesota. They cut all the old white pine down and they moved out west and started cutting down trees out there. But long story short, we think that basically with no pines around, the new host that really was sort of stepped in to be there for Suillus sabarius when the pines were absent were Quercus. And so they were able to basically stay on Quercus and perpetuate on Quercus. And that was the way that, again, we have seen this otherwise interesting and sort of anomalous host association. So this is a study that's published in a journal that's got some more details in it. I just posted this up here, but some work that again was done by Lotus and New and was published a few years ago in the New Phytologist. So I want to circle back now, and I want to tell you a few more stories about kind of what we know about slippery jacks and their host range. So the pine family is represented here. So that's what this phylogenetic tree, these are all the pine trees, and again, some representatives, not all the species, but you get these two big clades. You've got the pinoid clade and the abiatoid clade. And when we talk about the known hosts of Suillus, really there are kind of three main genera that are really known as kind of key hosts of Suillus. So you've got pines, again, both soft and hard pines, the 2-3 needle and the 5 needle pines. You got larches, and then we've got the Doug fir clade. In this case, in the U.S., really Suillus, and Pseudotsuga menzicii is the primary host in that genus. But as you can see, there's some other taxa that are present both in this pinoid clade, and then there's a lot of other. If you guys spend time out in the woods in Oregon or otherwise, other types of conifer trees that are often present in the same types of forests. So where there's Doug fir, you can find grand fir, right? And so Abies is present in many forests. If you move up into kind of higher elevation locations, you can find red fir and in mixed forests. And then of course hemlock's a big tree that you have out on the west coast there that we have some of in Minnesota, but not a lot. But Tsuga heterophila, of course. And so thinking about, could Suillus associate with some of these other hosts? From a evolutionary perspective, clearly, it can associate with hosts on both sides of in this case, spruces. And so that was a question. And then what about these abiatoid hosts? So things like fir, abies, or hemlock, Tsuga. So this was a interesting observation. This was up on, I believe it was Mushroom Observer, and my old PhD advisor, professor at the University of California, Berkeley, Tom Bruns, has this note. So they found this Suillus up in Alaska, actually, at Bonanza Creek near Fairbanks. And he says here, I'm puzzled by this Suillus. We found it in a forest of white spruce with paper birch and alder, but there was no pine within hundreds of miles and no larch. It has a prominent annulus and a gelatinous pilus, although it's dry weather. The tubes are radially elongated. I'm pretty sure I haven't seen this. I just bet a colleague a beer and hope that when putting it on Mushroom Observer, someone would recognize it and give it a correct name by day's end. And so this species, Suillus glandulosus, is actually an interesting species. If you look on iNaturalist and you type in Suillus glandulosus and look for records of where it's collected, of course, Alaska is way up here, and that's where Tom made those collections. But these are pictures of what some of this looks like. It really actually looks a lot like Suillus clintoniensis, although a little bit darker, and there's some differences that are important. And it is, in fact, a good, unique species that is not the same as what we call Suillus clintoniensis, Suillus gravelii. But it's here, and it's here throughout kind of eastern North America, but eastern boreal North America. And so the conifer hosts that we would think of are what Tom was saying, larch, so Larix laricina, we call it tamarack here in Minnesota. Black spruce, Picea mariana, although it could be white spruce, that would be Picea glauca if it was upland. And then Abies balsamia, so that's our fir that we had, balsam fir out here is another possible host. So this is a phylogeny, so new when that paper on Suillus, where we did a lot of work to kind of make sure we were thinking about which species were which and where globally, and then particularly these North American species. We can see here, if you look through the lists of all these different species, this indicates that we're associated with larch trees. So this is what the fascicles of a larch looks like. And you can see here that Suillus glandulosus, there's a collection from Alaska here, falls right into this otherwise larch associated Suillus group. So it seems like that would be the place that, we would look and think maybe larch is the kind of primary host, or the host that maybe is most normally associated with Suillus glandulosus based on this phylogenetic placement. So I was lucky to work with a fantastic graduate student who's still actually in my lab, Eduardo Perez Pasos. Is from Mexico, but he's been doing his PhD and done a lot of neat work. And so I want to talk about this work that we did with Suillus glandulosus here. And so what I mentioned before was this idea of a primary versus an alternative host. And so based on that phylogenetic placement, Eduardo and the folks we were working with thought, okay, well, Larix is probably the host that is most likely the primary host of Suillus glandulosus, and by that meaning that it has, again, chemicals that come out of its roots that allow the spores to germinate. And then possibly there are these other alternative hosts that we don't know about yet. And so that would be Suillus associated with maybe, again, the spruce, in this case, the black spruce, or maybe the balsam fir, again, from two groups that are, if we go back to, I'll show you the phylogeny in a minute, they're in places where we wouldn't expect phylogenetically for those to be the main hosts. So we set up a bioassay and we harvested this a couple of different time periods, and this is what we saw. So this is a lot of information. I can really I'll try to walk you through this, but basically what we're doing is we're taking seedlings that have been colonized with the spores of Suillus glandulosus, and so you've got larch on its own. You've got larch and, in this case, the fir growing together. You've got larch and the spruce growing together. And then you've got, in this case, the fir on its own. And then you're looking at what the colonization is on the fir when it's in the presence of larch. The spruce on its own. And then, again, with the larch present, what's the colonization on the fir? So what this graph is telling us is that there is, colonization really is, in the single species treatments, whether it's just larch, whether it's just fir, or whether it's just spruce. It's larch that is the one that is in fact the primary host for the species. So we see some colonization, and there's more colonization over time. So after about a year, we get more colonization than after 8 months. And then it colonizes this larch in all places. Interestingly, what we see is it takes a while to get colonization going, and that might be just a property of our bioassay and growth conditions, but we didn't see any colonization on either of these alternative hosts at the first time period. But at the second time period, we saw a pretty clear trend. We saw that when we had larch present, we could get colonization on the fir, and when we had larch present, we could get colonization on the spruce, but not when they were alone. And so that was important because that really suggests that, again, it's this primary host that is Larix that allows spores of Suillus glandulosus to germinate, colonize, form mycorrhizas, and then from mycelium it can grow out and it can colonize these alternative hosts. But those two hosts don't produce the same types of chemical cues, for spore germination. So here's what these root tips look like. So again, there's a little bit of variation in terms of how they look, and I'm not going to go into too much detail, but they've got these sort of white, kind of fluffy roots that are swollen, kind of look a little bit like hot dogs almost, but you can see that they make those same morphologies, whether it's on, in this case, the spruce or the fir, or on the primary host that is the larch trees. So the kind of take-home conclusions here from this one is that we see that there is actually the capacity of Suillus glandulosus associated both with the pine clade, which was the pinoid clade that we knew from the Pinaceae that had Suillus hosts, as well as the abiatoid clade. In this case, Abies is the first documentation here. However, both the Picea and the Abies, they really act as a different kind of host. So they can host if the fungus is growing in its vegetative state from mycelium, but if it's just spores landing in the vicinity, they don't have the same types of capacity to get those spores to germinate and grow and form mycorrhizas. And the colonization on those hosts is also something it's relatively low. They don't seem to really take off and love those hosts the same way that they do their primary host, but Suillus will grow on those hosts in the conditions that we've looked at. So I'll kind of wrap up with one last story that kind of thematically is very similar to what I've talked about so far. And so it's kind of testing the host associations, but this is Suillus punctatipes. So again, another mushroom from your neck of the woods. And so this is some work, and these pictures are actually taken by Noah Siegel, who's a fantastic mycologist or mushroom forager, mushroom identifier from Northern California, I think is his home base, but has done work all over Western North America as well as other places. And so he posted this record of Suillus punctatipes, but in talking to folks, including Noah, it seemed like people had collected this Suillus species in places where the known host, Doug fir, wasn't present. It could be in particularly high elevation locations, was areas where they were finding this Suillus and they weren't finding Doug fir, at least in the immediate vicinity. And so there was sort of this question mark of, could it be a different host that is maybe associated? And the possible hosts that were identified were in these high elevation forests, either mountain hemlock, and so that's Tsuga mertensiana, or maybe one of the AB species that was up in some of these forests as well. So this was work that Noah had done, and the collections had been done here in the Cascades, a little north of you in Washington State. So to remind you kind of who these other hosts could be, so I haven't talked about hemlock yet, but we did talk about the firs, and we showed that yes, it looks like Suillus glandulosus can colonize fir trees, true fir trees, the AB trees. But we'll look here and sort of talk more about it now. Again, the known hosts being the ones that are sort of up above, and this was all, the study was all done at one time. So I'm telling it to you sort of in a different chronology, but at the time, this ABs thing really had not been sort of worked out. So, sorry, left a couple of question marks from previously. So where does Suillus punctatipes sit in the big phylogeny of all the slippery jacks? Well, if you look and zoom in here, turns out there's a sequence of it that belongs in and associated with the 5-needle pines again. So we talked about the 5-needle pines earlier as the place where Suillus sabarius was close to. In this case, we've got Suillus punctatipes associated with, in this case, roots of Pseudotsuga menziesii. But interestingly, kind of from a phylogenetic perspective, falling in with not with the other Suillus that are associated with Doug fir, but with pine associated Suillus species and 5-needle pine in particular. So fortunately, we got some help from both Noah and then also from some other folks in the University of Washington. They were able to go out and make some collections for us in, again, the Cascades. And we did the same thing that we did in that previous study, where what we did was we looked at the roots of, in this case, conifers that had a morphology that looked like Suillus, and we did the DNA sequencing. And what we could say is that at all these different locations, we found, 18 root tips, and we could match and make sure they were in fact Suillus punctatipes. And we could see that, for out of those 18 that we knew were Suillus punctatipes, the host that they were associated with in this case was Abies. And we could see a similar pattern where in a couple other places we knew it was Suillus punctatipes, and the host they were associated with in this case was in fact hemlock. So this really helped us kind of nail down the idea that this particular Suillus species can in fact associate with both Abies and with Tsuga. So that was sort of interesting. We did do the same type of bioassay that we did before. So we used, and we kind of thought this was going to be our primary host, Pseudotsuga, and sure enough, yes, it was our primary host to get lots of colonization by, in this case, Suillus punctatipes on the roots of Doug fir seedlings. We used a 5-needle pine. For us, that's Pinus strobus here. We could see that there was some colonization of the 5-needle pine. So pines can act as what looked like a additional primary host. They can get some colonization, although not quite as much colonization here. We did not see for Abies really any colonization, maybe a teeny bit in the first harvest time, but really nothing in the second harvest time. So that was sort of interesting because it suggests that, yeah, we can find it on Abies out in the field, but Abies really doesn't look like, at least under the conditions we tested in the lab, a particularly great host for this Suillus species. So we tried one other treatment. It was actually a treatment that I featured just a little bit in the earlier talk with Lotus, but we decided, what if we actually got rid of the primary hosts? And in this case, the primary host we thought of as Pseudotsuga. And we tried this, and I wish we had done this. We didn't have all combinations of these treatments because it turned out that AB's seedlings didn't do so well. But we can see in the graph here that when you get rid of Doug fir, and you just leave, in this case, the other host around, pine, and it was eastern white pine, we don't see any difference in colonization. And so whether the primary host is gone or not doesn't really seem to change things. And the way we interpret that is that, again, the colonization doesn't really change in a significant way. What this means is that I think this means that when Suillus gets onto these alternative hosts, that it can actually stay on those hosts and do reasonably well. It doesn't have to have the primary host continuing to shuttle carbon over to it, even when it's on that secondary host. So the conclusions from this part of the study about Suillus punctatipes are that it can form both with pinoid and abiatoid hosts. So there is some sort of broad association there beyond just Pseudotsuga. Abies looks like it's a secondary host, so the spores don't germinate in its presence when alone. And again, this idea that primary hosts, once you can get colonization on an alternative host, it does seem like that can be sustained over time, even if the primary host still isn't present, which actually fits reasonably well with sort of our ideas about what we were thinking with Suillus sabarius being able to persist on oak, even though it started on pine. So this is a paper that we published with Eduardo and Ecology a couple of years ago. And again, here's the information if you're interested, and you can look and read more about it. And I've simplified a few things here for the sake of the talk, but you can find more details in the paper. So I guess I want to leave you here now and leave a little bit of time for questions at the end with kind of what are some kind of hopefully takeaway messages if you were to walk away from this talk and say, what do we think about kind of host specificity? And the first is that I think it's important to recognize that host specificity, I think what I've shown you today is a lot of this depends on ecological context. And so we can see for the slippery jacks that there is an association of slippery jacks with a bunch of different conifer hosts, not just the kind of iconic classic ones, and even some angiosperm hosts. This could be important with shifting climatic conditions, so novel host associations. As those become more common, maybe we're going to see some changes that are different in terms of the associations that are out there. And that might be important actually for thinking about how these forests are going to respond. That said, we just don't see out in nature these associations very commonly. It's not like we see, particularly for Suillus, it often on these alternative hosts. And so we've been scratching our heads a little bit, and we kind of have 4 different reasons, and I'm listing them down here. But why are these alternative associations relatively rare? And I think part of it has to do with the fact that unlike above ground, when you go down on the root tips, there's actually not a whole lot of colonization. Suillus has a interesting way of pulling a lot of carbon out of these trees to make these big, beautiful mushrooms. But below ground, Suillus is not super abundant. We don't usually find a lot of these hosts in kind of direct proximity where the roots are right next to each other. And that can be due to things like different forest successional stages or different, elevational gradients and in terms of latitude of where hosts are. So maybe that's part of why we don't see, for example, Suillus on these alternative hosts that often. If they are both present in the same place, there are lots and lots of other mycorrhizal fungi in these forests that are colonizing the roots, and so there may not be a lot of free tips for Suillus to move around on across these different hosts. And we actually know that Suillus is not a particularly strong competitor for the root tips in various different experiments that we've done. And so it may actually kind of lose out in its place in some of these communities. And because it's not an aggressive competitor, it really can't establish itself well on other hosts. So these are all sort of open questions that we would love to continue to kind of do research on and speculate about, but ways to kind of think about this. So interesting flexibility in host association, maybe more unexpected than we would think, but ecologically, I think there's actually some good reason to think, yes, generally, this kind of pattern of which hosts they're associated with holds up reasonably well. So I just wanted to finish here with some acknowledgements, and I actually want to acknowledge someone who's from Oregon. I doubt he's listening to this conversation, but it's a person who was really important in my own thinking and actually a mentor to me when I first moved up to Oregon as assistant professor. Randy Molina. So Randy was a longtime employee and actually leader of the U.S. Forest Service mycology group at Oregon State University, and had an adjunct faculty position there as well. Randy did all kinds of great work on host specificity and really kind of, I think, opened this whole avenue and line of research up with lots and lots of cool studies throughout the 80s and 90s. And a lot of the work that I've done, both with Suillus to some extent, but particularly with Rhizopogon, I didn't talk much about today. A lot of that is built on the foundation that Randy and colleagues built. So I want to give him a special acknowledgement there. Been super lucky to work with some really fantastic people. So like I said, New Wynn's a professor at the University of Hawaii. Rita Spogles is a great collaborator and colleague out at Duke University. Renee LeBouf and Noah Siegel were collaborators on our project with Eduardo. Students, Lotus, Eduardo, some undergrads that have been in the lab who've done all kinds of great work. And then some funding that has supported this research has come from the National Science Foundation. So with that, I'm happy to wrap up, and hopefully I've left a little time for questions here, but I can't see anything with the way that Zoom works. So I'm gonna stop sharing, and maybe then I can see what folks, if they have questions, I can answer them. So thank you very much.
Well, thank you, Dr. Kennedy. We'll see if there have been any questions that come in. Sandy, have we got any questions
Yes,
in the chat?
we have quite a few.
Okay, I'll let you voice them then.
I don't know if you prefer me just to tell you the questions or put them in the chat, or
Either way, whatever's easiest. Sandy, if
okay.
you want to
The short ones I'll just ask. There's some really long ones I may move over to the chat for you.
Sure, great.
So let me see. The first one is just, I think it's more of a, Suillus americanus, is that the one that causes the skin irritation? You know anything about that?
I have heard that actually, and
Okay.
I don't know whether it's not a particularly slimy one. I've heard that there is some that do have that reaction if you handle them. And I have fortunately not. I've handled a lot of Suillus. I haven't had that happen to me, but I think folks that have any kind of hypersensitivity to mushrooms, that is true. I don't know that that's unique to Suillus americanus, but yes.
And that was from, I don't know if you'll recognize these. We don't have any names. That was from Blue Froggish one. Blue Froggish one. I don't know.
Don't know that. So the closest relative to Suillus americanus that you'd find out in Oregon, you'd probably find up in the high elevation. It'd have to be really pretty high elevation. There's Suillus sibiricus, but that's a western species that would be associated with some of the pines up at the tops of the mountains in the Cascades.
Okay, and also from the same person is, wonder what role soil bacteria plays in colonization of these different hosts.
Yeah, no, that's a great question. We haven't thought a lot about that in our case. I assume that there is definitely some important interactions that are part of that equation. There's a whole story. There was a French research team that did a lot of work on these mycorrhizal helper bacteria. So particularly some in the genus Pseudomonas were well known to be facilitators of colonization. And so there's some, kind of particularly fascinating things that we could think about. There are bacteria that live not just on the outside in the soil, but there are bacteria that live on the inside of hyphae. There are endohyphal bacteria that might also be important. We used experimental conditions where we were careful to just control which fungi were present. So we sterilized the soil before we added spores. But we grew them in, greenhouses that were not 100% sterile. And so I'm guessing there might have been some bacteria in there, but not something we got specific data for in this case. But yes, that is, I think, a known part of the association. There's also some fascinating stuff with bacteria associated with the mushrooms themselves. There's a whole consortia of mushrooms that when they die, they get gobbled up and eaten by all kinds of bacteria and fungi that are important in that way too.
Okay, the next one is like 5 parts, maybe. I'm not sure if it's question, comments, a little bit of both.
Okay.
I put it in the chat. If you could, do you see the chat? Can you click on the chat?
Unfortunately,
I know. Trying to find anything on Zoom is a challenge.
You can,
I'll
unfortunately,
go ahead and start.
Yeah.
So the first part is, this makes me think of studies of there's a lot of generalists by
Endomycorrhizal.
colonizing brassicas, usually incompatible, even in the same soil, when an important nutrient is scarce, like what, nitrogen and potassium,
Sure.
NP.
Yep.
So
Yep.
with the
Yeah.
right motivation, perhaps a robust endo or ecto can be encouraged to leap host species.
Mhm.
And
Yeah.
it keeps going.
Okay. I mean,
Maybe appearance
go ahead.
speciation can be stimulated under novel local ecosystem conditions initiated due to climate change to help host species better adapt to the new conditions.
Yeah, so I mean, I think that honestly, I hope one of the takeaways from this talk is that, yeah, again, environmental context matters a lot, and so I think there is some interesting studies. So brassica is, one that is classically non-mycorrhizal, but it does have these interesting associations with some fungi in its roots. I haven't kept up with that literature as much as I probably should, but yes, there are some interesting associations. I don't know how much they are sort of nutrient context dependent. It is a fascinating way to think about this. There's actually a lot of variation across Suillus species in kind of how much they use, for example, organic nitrogen versus inorganic nitrogen. And I think that could lead to some interesting questions to kind of think about along those lines. In terms of, thinking about climate change and host association, we have done some work with some other Suillus species thinking about how they might respond to drought conditions in particular. And there is some interesting acclimation that they seem possible of doing with regards to dealing with drought-like conditions. So their gene expression patterns under heavily droughted conditions. Whether that would, form a novel association. I guess the one thing I should emphasize is that, again, most plants and most trees are associated with, if not tens, hundreds of different species at the same time. So I guess, I wouldn't be surprised if environmental selection did drive some capacity for maybe new types of associations, but I don't know. I'm posing that as a question that I think is important to think about, and I do think that having these associations can be really key to handling changing environmental conditions. But exactly how they drive speciation, I think that would be just a lot of interesting speculation. I'd love to hopefully bring some science to it somehow.
Okay, there was okay, the same person went on to say it would be interesting to try to initiate new associations. Wait just a second, things are moving here. Where shifting field conditions do not currently exist to stimulate these, perhaps transplanting a colonized tree to a new environment.
Yeah, so that's been done, to some extent in timber industry, right? So people have tried to plant pines in the southern hemisphere where they weren't for a long time, and they had very little success planting those pines for a long time in particularly Australia and New Zealand until they started bringing in some of the soil, and it turns out that some of that soil had mycorrhizal fungi in it, and some of those were Suillus species. So those are ones that I think show how important the association is for establishment. It's a little bit of a double-edged sword. There can be invasions of pines in particular that can be facilitated by Suillus, and that can be problematic in southern hemisphere ecosystems. But I agree with sort of the idea that we should think about these across some set of environmental gradients. And the good thing is there's lots of interesting experiments that are being run in different locations with altered precipitation levels, with altered warming levels, etc. And I think those are ripe for the exact kind of experimental approach that this person's proposing, right? Is what happens when we put together these symbionts in these novel settings? Does it help them persist or not? So I like a lot of the line of thinking there. It's just a question of trying to do that science and see how it plays out.
Okay, here's a question from Grace Pugh, longtime CMS member. Is there any indication of exactly when the association shifted based on the genetic tree, or are signs pointing to this being a very modern shift?
Yeah, that's a great question. So, we're seeing these host shifts across lots of different Suillus that are not particularly closely related to each other. So I think this is something that suggests that it's not as if it's a kind of basal phenomenon that happened, and then sort of allow for these shifts later on. The stuff that we've done, I think, is really on sort of relatively shorter timescales. I think, the ancestral Suillus host from reconstructions we've done from work using like genomes of lots and lots of Suillus species looks like it was large, actually, and probably somewhere in Asia was probably the original sort of location for where we had a lot of Suillus initial diversification. And then from there, it's branched out into other host associations, but there's been some moving around within those. So I don't know if I'm answering, Grace's question super clearly. I think the stories that I have told you today, the science that we've worked on, have suggested there's this kind of interesting ecological timescale flexibility, the deep evolutionary history of host switching. That is something that, sort of is seen. It looks like there are lineages that have associated with white pine or with red pine, actually the 2-needle versus the 5-needle pine. And that's happened a handful of different times throughout the sort of evolution of the association. But it's not something that is super well rooted in, happened once a long time ago and that was it.
Okay. Here's a couple from Dylan. Not sure if it's one question or more. Have you identified the mechanism by which these species have a stronghold into an ecosystem from the ghost mycorrhizal past used to spread in new growth of this secondary host species? Is it a genetic alteration? Additionally, is there a duration of this phenomenon takes place, or once in place, can it last indefinitely?
Yeah, so it's a good question. Oh, sorry. And I don't think it has anything to do with genetic alteration. So I think that, it's the same Suillus, in this case, Savarius, that's on oak, that's on pine. They came out of the same mushrooms. There's really nothing that suggests that there's some genetic anomaly that has now allowed it to colonize oak and do better there. So I think that's not what's going on. I think that, again, it comes down to basically the local environment it's living in, sort of the needs of the host, and those kinds of factors are probably most important in allowing it to persist. If it's able to, grow underground and colonize new roots, it seems like it can happen for a long time. And so I think the question, the second question about how long you could have this, I think some of our data suggests that once you get onto an alternative host, as long as there are alternative root tips that you can continue to colonize, that you can get carbon from, and there are nutrients that you can continue to get out of the environment to trade for that carbon, I don't see any reason why that would stop per se. Certainly trees, are going to live a certain lifespan, but in that vegetative state, it looks like there's much more capacity to crossover. And so again, it would depend on the particular successional trajectory of the forest to make those predictions, but it does look like it can hold on for a long time. The white pines were cut out of the Lake Alexander woods, definitely, back in sometime in probably the late 1800s, and we were sampling in 2014. There was a few white pines, like I said, at the site, but really none of the big ones that were left. And some of those oak trees were actually quite large that we were sampling, so they've probably been around for 80, 90 years. So how that switchover happened, and then did it jump from pine to oak, and then was it the same oak that we sampled 80 years ago that was next to a pine? I don't think so. So I think it's been going on for a while. But yeah, good question. I don't know that we have exact data to answer it.
Okay, I just asked if there was additional questions. We have gotten observations that it is very cold where you're at.
Yes, there is no
That person
slippery
was
jack.
very happy they're sitting in California.
Nice. Yeah, there's no slippery jack mushrooms out in Minnesota right now. I can tell you that.
I must admit, I picked, so it's the first day of winter here, and I picked some chanterelles today.
Oh, lovely. Makes me happy that somebody's picking mushrooms right now. That's great.
So I have a non-technical question about Suillus and Rhizopogon,
Yeah,
just from a non-technical person's point of view.
sure.
The recent switch to bringing the hypogeous Rhizopogons into genus Suillus. What's your viewpoint on that?
Yeah, so I mean, I think there are so I think they are good distinct genera in the sense that I think Rhizopogon does not need to be subsumed into, in this case, Suillus. They are sister genera. They are closely related to each other. And when we look at the phylogenies and we work hard to figure out, sort of what's going on there, it does look like, Rhizopogon and Suillus kind of fall out very, very close to each other. But I don't think we should change all the name of Rhizopogon to Suillus. There is a lot of hypogeous fungi that are close relatives to mushroom-producing fungi that should actually take those names. Interestingly, there is a hypogeous Suillus species that a graduate student in Joey Spatafora's lab at Oregon State has found. And there's a few other weird Suillus, Gastrosuillus, and a few other things that are strange secosioid sort of intermediates, so not truffle, but also not really looking like a regular above ground sort of open pored mushroom as well. And she's found that in Oregon. So you guys should try and get Carolina to come down and tell you guys some stories about that. But that looks like a true truffle. That is in fact actually it is a Suillus by genetic association. So I would not put Rhizopogon into the genus Suillus from a taxonomic perspective. But yes, there are definitely other species of truffles that really should have the name of their mushroom forming counterparts based on what we see from their genetic data.
So I have another question along that same lines. We had a speaker years ago, and I'm failing to remember the person's name, but they were studying the soil biology of tuber species, both in Spain and here in Oregon.
Nice.
And they had reported on their work in Spain because the work in Oregon had not been published yet, but the species there in the calcareous soils, the species of tuber were altering the pH of the immediate root zone of their host trees to make it more basic. Basically root pruned to give them more space to attach. Do you see anything like that pH change happening with the Suillus or Rhizopogon?
Yeah, no, it's a great question. I think I know who you're talking about. I'm not remembering his last name. Carlos is his first name, but he spent some time in Oregon and had done a lot of work on truffle in Spain. But that was a good while ago. Anyways, so that brulee formation that you see with tuber is really relatively unique as to what I know. A lot of it has to do with actually some chemical volatiles in addition to changing the pH. But we don't see that same pattern with Suillus and Rhizopogon. That said, we have not looked particularly hard at that question in that particular way. And so I can imagine that there's some modification. All fungi, when they are accessing, nutrients in soil, they're often pumping out lots of, protons. And so, they tend to acidify their environments to some extent. But no, I don't know of any equivalent story for Suillus and Rhizopogon as that tuber story. It's a just fascinating part of the biology of that genus that is unique, I think. But yeah, modification of pH is just something that's I think it's generally seen, but not at the scale that you see it for tubers. So
nice. Well, unless Sandy, do we have any more online questions that have come in since we last checked?
Nope.
Well, then I think we're at the end of our time with Dr. Kennedy. Thank you so much for speaking to Cascade Mycological Society. And as always, with all of our presenters who have presented via Zoom, we invite you to come visit us at some time in the future and be our guest here in Eugene.
I would love to do that. Thank you again for the invitation. It was really nice to meet you. And yeah, enjoy the mushroom hunting. I hope you can get out and collect some more chanterelles. And one of these days, I'll get out west and come down and visit and say hi in person.
Definitely do. Happy solstice, and hope to see you again in the future.
Sounds great. All right. Take care, everybody. Happy holidays.
Chris, do you have announcements, correct, Chris?
Yes, we do. So next month's speaker, January 2023, is Buck McAdoo. And he's got a book out on his, he's been writing a column for his Mycological Society, and it's about the different species he's ID'd and encountered over his years of mushroom hunting. And we'll be visiting with Buck via Zoom as well on the third Wednesday in January. So hope to have you tune in then, and happy holidays to all.
I guess that's my cue to stop.